Welcome back to the chapter, students, and in this module we shall be studying the process of our DNA technology and the production of insulin using our DNA technology. Now let us move ahead and begin with the module. So here I have DNA 1 and I have DNA 2. Now when I combine these two DNA, I get a recombinant DNA. So these are two different DNA from two different sources. Now let us see how do we do that. Here I have an enzyme which is called as restriction endonuclease, or which you have studied as molecular scissor. Now this restriction endonuclease enzyme will cut my DNA and give me a pattern or a cleavage pattern which is called as sticky ends. Now remember that these sticky ends are single stranded overhanging DNA after the double stranded part of the DNA. These are the sticky ends. Now what happens with the sticky ends? These sticky ends will help me to form the recombinant DNA molecule. Now when I combine these two DNA molecules that have been cut with restriction endonuclease, they are able to undergo complementary base pairing. However, even after undergoing complementary base pairing, there is something which is a break or a nick in the DNA molecule. Now which enzyme will seal this nick? Yes, it is DNA ligase. So here I have DNA ligase, which is sealing the nick or the break in the DNA strand and giving me a complete DNA molecule. So here I have a complete DNA molecule which has sequences from both the sources, and this new DNA molecule is termed as recombinant DNA or the chimeric DNA. So this is how we carry out the process of our DNA. Now what do we do with this recombinant DNA? We allow this recombinant DNA to undergo cloning so that we can get multiple copies of this recombinant DNA, and this will be achieved in vitro with the help of polymerase chain reaction or PCR. Whereas in vivo, it will be achieved with the help of unicellular prokaryotes like E. coli, unicellular eukaryotes like yeast, and mammalian tissue culture cells. So this is the process of our DNA technology. Now let us move ahead and take a look at how we produce insulin with the help of our DNA technology. So here I have a human cell from whom I have removed or isolated my DNA. And what do? I cut this DNA with the help of restriction enzyme. Why do I do it? I do it to isolate the gene for insulin. Now I have here E. coli, and what do? I isolate the plasmid vector from E. coli. And next, what will be the step? Yes, I will cut my plasmid vector with the same restriction enzyme that has been used to isolate the gene for insulin. Now I have another enzyme which will help me form a recombinant DNA molecule, which is nothing but DNA ligase. So with the help of DNA ligase, I get a recombinant DNA which is nothing but my plasmid vector or the cloning vector carrying the gene for insulin. Now what do I do with this recombinant DNA vector? I insert this recombinant DNA vector or introduce it into E. coli cells. And what do I do with these E. coli cells? I allow these E. coli cells to multiply, and hence I have a number of E. coli cells showing me the cloning vector with the gene for insulin. So finally I have a population of insulin producing E. coli cells. So this is how insulin is produced using our DNA technology. So in this module, we studied the process of our DNA technology and we studied the production of insulin using our DNA technology. Whereas in the next module, we shall be studying certain therapeutic products made by our DNA technology. So till then, study well, Take care and bye bye.